Hello everyone, my name is Yile Liu. Today I will be presenting the WI38 cell line, the biological concept, medical impact, and social relevance of it. This talk will explore how human cell line originated, how it has contributed to medicine, and how it connected to broader ethical and social questions. So here's our roadmap. First, we'll revisit fundamental biological concepts such as cell division and what human diploid cell strings are. Next, we'll divide into origin and characteristics of the WI38 cell line. There, we'll examine its medical impact, especially its role in vaccine development. After that, we will explore the historical and social relevance, including ethical concerns and public policy implementations. Finally, I will offer some personal reflection on why the WI38 matters to me and our society. So here's the basics of cell division. To set the stage, eukaryotic cells divide via mitosis, which is a process of chromosome replication, alignment, and segregation into daughter cells. Human diploid cells have two complete sets of chromosomes and represent normal, non-cancerous human tissue. Before 1960s, most cell cultures used for research or vaccine production were either primary or cancer-derived. What was lacking was a well-characterized normal human diploid line that could be reliable used for larger scale culture. So in the cell division, here is interphase, which include G1 S phase, G2 phase. In G1 phase, it's basically cell growth. In S phase, it's basically replication of the genetic materials, and for G2, it's also replication of proteins and other preparations for the mitosis phase. For M phase, the first one is prophase. As you can see here, the prophase, the nucleus started to to become more condensed and chromatins formed, and in metaphase they lined up here in a line, and in anaphase they separated, and each daughter chromosome go to one pole of the cell, and in telophase it eventually comes to two cell by cytokinesis. So in 1962, Dr. Leonard Hayflick and Dr. Paul Moorhead discovered this WI38 cell in Westard institution. So WI basically means this institution's name, and it is in Philadelphia. And it is the 38th cell string successfully derived. The characteristics and maintenance of it. So WI38 is a normal human diploid fibroblast line. It means it can retain 46 chromosomes and have a finite lifespan. So, because it could be frozen, swallowed, and constantly passage while remaining well characterized, it became ideal for virus production, such as it can make vaccines. And unlike many immortalized or tumor lines, it retains many normal cell behaviors and offers a safe substance for viral vaccine. So it is used globally in controlled laboratories, and it should be stored in cryopreservation. And in the next slide, we will talk about medical impacts. So one of the major scientific legacy of WI38 is that the demonstration that normal human cells are not immortal. Like Hayflick discovered this cell eventually stopped dividing, a phenomenon now known as the Hayflick limit. This discovery opened up research into telomeres and cellular aging, and also normal human cell differ from cancer cell. WI38 does bridges cell biology and aging research. It also provides a source for vaccine research, because before that, many research use animal tissues which are contaminated and not really relevant for human to use that later. And now it is clean and standardized because we can use WI38 human diploid cell line. And here's a case study of how it applied into medicine field. So the vaccine is called rubella RA27-3 vaccine. It was developed by Stanley Plotkin in 1969, and the virus was attenuated through serial passage in WI38 cells. It is safe and effective and now part of MMR vaccine, and it prevents congenital rubella syndrome worldwide. This breakthrough challenge of global public health. Here's the historical context. So in early 1960s, there was primarily kidney cell used for vaccine were repeatedly contaminated. WI38 arrived at a critical moment. A normal human cell line, thoroughly tested, safe, and scalable. It is available accelerated vaccine development at a pivot period in public health. And Hayflick challenged the immortal cell dogma, as I talked before. WI38 provide a normal human cell that can have a limited lifespan, and this make vaccine production shifted from animal to human based. Here's the ethical concerns and controversies. The story involves a key ethical dimension, the fatal origin of WI38 triggers debates about use of abortion-derived tissue in research and vaccine production. Some religious and ethical groups still question this use of this in cell lines. On the other hand, many bioethics comments and the Vatican's pontifical academic have judged the use of mortal permissible when the alternative options are limited and public benefit is high. So it influenced modern bioethics and informed consent policies. WI38's legacy influenced policies, informed consent procedures for tissue donation, fetal tissue research guidelines, and banking for cell lines, and vaccine regulation protocols. This showed how a cellular research breakthrough can have a ripple effect into global health policy and ethical overlay. It also influenced NIH and FDA regulations, enable global vaccine programs through safe human cell culture. Here's my personal reflection and summary. So I think WI38 medical benefit affect all of us. Beyond science is at a intersection of identity, trust, and science. It provoked question how we value normal human tissue in research, how public knowledge of vaccine development affects trust, and how scientific decision reflects social values. It invites reflection on whether scientists program can and should proceed with fully transparency and ethical regard. So as a summary, WI38 events offer understanding of cell division and aging. It enables safe vaccine that save life worldwide, and it sparked ethical reflection on use of human tissue. It continuously influenced medicine policy and public health. So we should be grateful that they discovered WI38 so we can have a healthier life nowadays. And here's the reference I used from online sources. Thank you for listening.